instead of killing cancer cells, we could just like reprogram them back to normal.
Yeah, it's like flipping a switch.
Yeah.
And we've got two sources to dive into today.
Right.
A scientific paper straight from Advanced Science titled "A Factor Landscape Analysis Reveals a Reversion Switch in the Transition of Colorectal Cancer," and a news article that summarizes the findings called "Cancer Breakthrough as Scientists Discover Cancer Cells." So, ready to jump in?
Let's do it.
Scientists in South Korea are exploring this revolutionary approach.
Their work centers around this system called REVERT. They used to analyze colon cancer cells. But they weren't just looking at any cells, they zeroed in on that critical moment. The cell transitions from normal to cancerous.
Also, they're focusing on that tipping point, like that split second before water goes. It's not quite liquid, not quite steam.
Precisely. And they use these lab-grown mini tumors called organoids. And they analyze them at like a single cell level to capture that exact.
Wow.
And to track the lineage of these cells as they evolve, they factored in CNVs, which are essentially unique genetic markers.
So like a cellular fingerprint, helping them trace the lineage of each cell.
Exactly. This allows them to create a detailed model of all those complex interactions happening with the cells, almost like mapping genes as circuits on a motherboard.
Okay, that's a really wild way to visualize it.
It is.
So how does all of this tie into actually reversing cancer?
Well, the researchers used their model to simulate what would happen if they like different genes, like which they were looking for a way to reshape the cellular landscape.
Okay.
So imagine a map with valleys representing different stable states a cell can exist in.
So in a cancer cell, the map is skewed towards like a deep, inescapable cancer cell.
Precisely. Yeah, their simulations revealed two genes, MYC and YY1, that seem to act as potential switches, but directly targeting these could be really risky. It could be like throwing a wrench into the entire cellular machinery, and it could potentially cause some serious side effects.
So they needed a more precise approach.
And here's where it's really genius. They hunted for a gene regulated by both MYC and YY1, and they ultimately landed on USB7.
USB7, okay. And what's so special about this one?
Well, USB7, with specific protein 7, is like a cellular garbage system. It tags proteins for destruction. USB7 in those colon cancer organoids actually slowed their growth. Even more exciting, it shifted their gene expression back towards a more normal profile.
So they found a way to reprogram those road cells? That's amazing.
And based on analysis from the DevMap and L1000 databases, huge repositories of information about gene activity, USB7 inhibition seems to affect cancer cells more than healthy cells.
So potentially fewer side effects. That's huge. But we're talking about lab-grown mini tumors here. How does this translate to, real-world cancer?
That's where the advanced science paper really digs deep into the molecular mechanisms at play. Inhibiting USB7 appears to downregulate key cancer-related pathways.
So it's like they're starving those cancer cells.
Yeah.
But how can they be sure that these changes are actually leading to a reversal of the cancerous behavior, not just slowing it down?
Well, they didn't just look at gene expression. They also examined the organism's growth patterns and even the physical appearance under a microscope. What they found was remarkable. It also caused them to start looking more like a colon cell.
But not just acting differently, but actually looking different too. That's pretty neat.
It is. And remember those potential side effects we talked about with targeting MYC and YY1. More promising if they USB7 inhibition seems to be much more selective for cancer cells.
So it's like a sniper taking out the enemy while minimizing damage.
Their analysis of the DevMap and L1000 databases indicated that 7 has a much more significant impact on cancer cells than healthy ones, which could mean fewer and less severe side effects.
Okay, so they potentially found a way to target and reprogram cancer cells. But is this really the magic bullet we've all been waiting for?
Well, it's important to remember that this research is still in its early stages. Still a long way to go. This could become a treatment available for patients.
Right, not quite ready for prime time. But it's definitely a fascinating glimpse into the future where cancer treatment might be less about the
That brings us to a question. What other diseases might be susceptible with this kind of like switch flipping approach?
Oh, now you're getting me thinking. this be applied to Huntington's, Alzheimer's, even aging itself.
The possibility. But before we get ahead of ourselves, let's delve into the specifics of this USP discovery. What makes it so promising?
You're right, let's break it down. How did they even pinpoint USB7 as a potential target in the first place?
It all goes back to those two key genes we talked about earlier, MYC, YY. Remember, direct targeting those could lead to a cascade of